Hello. I would like to thank to the organizing committee for giving me the opportunity to present our work. So back in 600 BC, people recorded that ants were attracted to the urine of diabetic patients. And that was due to the high levels of glucose present in the urine. Later on in the Middle Ages, physicians diagnosed diabetes by tasting the urine. And it was also nicely put by the physician Thomas Wheeler back in 1674 as a wonderfully sweet, as if it were imbued with honey or sugar. So today I'm going to talk with you about tasting urine, but not with our tongue, but with electrochemical sensor, and how we can understand from the measuring those urine or biofluids about our medical condition. Redox molecules are present in any different types of mechanisms in our body. They have an important role on the regulation, communication of different physiological pathways, either endogenous metabolism or exogenous impact. For example, there is kind of redox homeostasis and the oxidative stress have an effect or a major effect in different types of brain diseases and various cancers. Current approaches to measure these redox molecules are based on a benchtop, costly setup such as mass spectrometry, microplate readers. They require various pretreatment steps such as freezing, thawing, adding different types of label reagents, transferring the sample from where it was taken from the patient into the analytical lab, and all these kind of steps actually changes the level and the redox state of the molecules, therefore decreasing the quality of the redox information, meaning that the composition of the molecule that was when it was taken from the sample from the patient already changed when it's being analyzed. Therefore, it's one of the different solutions that can rapidly analyze redox biomolecules that basically can move the lab to where the sample is taken. So I'm a bit biased, but electrochemical sensors are a very optimal solution to measure redox molecules, mainly due to the electrochemical activity. So, however, when you try to use a electrochemical sensor to directly measure in biofluids, there's a high limited sensitivity and specificity due to two major challenges. So when we measure redox analytes, we generate a specific electrochemical fingerprint. However, when we try to measure this analyte directly in biofluids without any pretreatment steps, in most cases there are also other redox analytes that we want to measure, and in many events they also generate an overlapping electrochemical signals. So we need a way to differentiate those signals. And that's challenge one. The second challenge is that there are also other type of redox molecules abundantly present in the biological fluid, and in many cases they actually have a higher concentration, so they actually generate a masking electrochemical signals. So we need to think about new ways how to transduce this kind of biomolecular information using electrochemical sensors. So in this talk I'm going to show you two main approaches that my group is working on. One is how we can use the chemical sensor to directly detect a specific redox analyte in biofluid, and also how we can actually detect an N number, one redox analytes. So for the first project I would like to show you is related to schizophrenia and the burden of monitoring the treatment. So the more than 1% of the population is being diagnosed with schizophrenia, and those that are being diagnosed require lifelong treatment with antipsychotics. Now there's this medication called Clozapine. Currently the most effective medication available for schizophrenia, but it's dramatically underutilized due to the monitoring scheme when patient need to in order to get a prescription, need to leave their home, go to the doctor, get the blood drawn, being analyzed in an analytical lab, go back home and wait for the result to come back, and then they can get their prescription. So many patients actually stop taking the medication, and when working with psychiatrists, they actually provide a kind of solution that if you can bring the lab to the place it was the sample is taken, such as in a doctor or even at home. Current state of the art for clozapine detection using electrochemical sensors are based on the inherent behavior of clozapine that can be electrochemically oxidized or reduced. Now here you have this kind of scheme that the y-axis show the work related to measuring clozapine in a milliliter volume samples or macro volume samples, or in a microliter volume samples, more related to the finger prick samples. And the x-axis we have measuring clozapine buffered solution, serum, which requires some pre-treatment steps, and eventually whole blood without any pre-treatment steps. So many work show how you can detect clozapine directly in diluted serum, such as the work by Tamari using magnetic particle modified electrodes, and also the work by Kang et al. Using electrode modified with graphene, and here without any dilution measuring clozapine directly in serum. Now this two work, current state of the art, they showed how they can measure that in a macro scale, and also with some kind of pre-treatment steps. So potentially, if we have a way to measure the clozapine directly in microliter volume samples, directly in whole blood, we can enable this better analytical way to measure molecules directly at the point of care. So previous work showed that the main masking and overlapping signals are uric acid, ascorbic acid, and cysteine as the interfering molecules. And our approach is that by modifying the electrode with electrocatalytic material carbon nanotubes encapsulated in the biopolymer chitosan, we could have an electrocatalytic effect on some of the redox molecules that now will shift or decrease the activation energy and shift the potential peak of some of the molecules, revealing the signal from the clozapine and improving the detection performance. So for that, we microfabricated a chip that has 6 electrochemical reaction chambers. Each one has a working count and reference electrode, and we use electrode deposition methods to modify the working electrode with the Chitosan-Carbon nanotubes biometrics directly in the working electrode. So the first part, we wanted to ask ourselves, what is the contribution or what is this electrochemical signature we measure from each one of the redox molecules, from clozapine and the main interfering molecules, and how the signal is affected by the modification with the Chitosan-Carbon nanotube. So what you can see here is the signature that the voltammograms from a bare and a Chitosan-Carbon nanotube electrodes measure with the clozapine by itself, L-homocysteine, ascorbic acid, uric acid, and dopamine by themselves in buffer solutions. And what we want to look at is, okay, we have the signature from a bare electrode and how it was shifted, the peak was shifted when we introduced the Chitosan-Carbon nanotube. And it was done for each one of the interfering molecules, or each one of the redox molecules. So in this table, I would like to look at the 2 columns that are circled with a red line, how the peak shifted between the bare and Chitosan-Carbon nanotube and how the current increased or decreased. What you can see is very interesting that other than uric acid, all the other redox molecules, they actually shifted the potential into low potential. So we can see that there is some kind of electrocatalytic effect of the carbon nanotubes on the molecules other than the uric acid. So we can see that by introducing Chitosan-Carbon nanotubes, we already overcoming the challenge of masking electrochemical signals from the main interfering molecule that has a high concentration, which is uric acid. So now what happens is the question about all the other redox molecules, and for that we tested how the electrochemical signal of clozapine with the calcium-carbon nanotubes is affected when we introduce the interfering molecule. So how the signal either increased, decreased, or shifted. And here we can see another table. And again, I would like you to look at the columns that are circled with red. And here we can see how the peak potential of the clozapine shifted in the presence of the interfering molecule, or how it increased or decreased. And what we can see, that the main effect was on the amplitude of the current, and obviously uric acid, which has the highest concentration, increased the signal, but we already shifted it. And ascorbic acid, and then we in the second highest interfering molecule was ascorbic acid, then dopamine, and then homocysteine. So by that we further understand how the electrochemical signatures of are affected by the different presence of different redox molecules. We also tested the repeatability, reproducibility, and storage stability. We showed higher that was able to sustain the signal in serum for 9 repeats with high reproducible signal, and also for 8 days in storage in dark and dry conditions. So the big question is, can we use this sensor to measure clozapine directly in whole blood? And for that we tested the electrochemical signatures. We took whole blood for volunteers, and in 5 minutes we spiked it with clozapine and measured the signal. On the left hand side measured with bare electrode, and on the right hand side with the calcium-carbon nanotube. And we built the calibration curve by looking at the signals at 0.2 volts, which before previous slide I showed you that it was the signal from the clozapine with the calcium-carbon nanotube. And we achieved a very good limit of detection of 0.5 micromolar, and that's very relevant to the clinical threshold when psychiatrists told us we would like to know if the levels of the clozapine in the whole blood are higher than 1. So we actually got 2 times better limit of detection than what the clinical threshold had. And this is the first demonstration of clozapine detection directly in microliter volume samples of whole blood in the required clinical range. And that's work, you can read more about it in the Advanced Healthcare Material paper that we recently published. The second question, I hope I convinced you that you can use a electrochemical sensor modified with modifications, coatings to measure a specific redox analyte directly in biological fluid without any pretreatment steps. However, the more and more research we see that a single analyte is not enough to have information about the medical condition of the patient, of the person, and it requires a profile of different redox molecules. So that the approach that we are working on, that we propose, is actually inspired by the best analytical device we have in our body, which is the tongue. So how we use the tongue to taste different samples, or for example wine. Our tongue has an array of bioreceptors. Each receptor generates a signal which is a bit different than the other one to the sample itself, generating a fingerprint that our brain can do the signal processing and decide about the flavor or the results of the sample, what is the sample itself. So our approach is using an array, is to differentiate or quantify multiple redox molecules directly in the biofluid by using an array of electrode modified with different modifications, generating data that now they are partially selective to the different molecules we have in our sample. They will generate a set of cross-reactive electrochemical signatures that now we use chemometrics or machine learning algorithms to learn or to identify a specific fingerprint for each one of the molecules. And now by measuring the samples, we can directly quantify the levels of the molecules by using the different models. So we tested our system on a proof concept study on the ability to differentiate the signals of norepinephrine and dopamine, which are two different neurotransmitters, directly in urine. And we chose them because their signal is highly overlapping. The first part, we wanted to see if what is the partial selectivity, if the electrodes are more selective to dopamine or norepinephrine, because eventually we want to have partially selective electrodes. And for that, we measure the dose response characteristics for either bare, Chitosan-modified, or Chitosan-Carbon-modified electrodes, and we calculate the partial selectivity. We showed very interestingly that, for example, the bare electrode was more selective to norepinephrine than dopamine, and also Chitosan-Carbon nanotubes, but the levels of the selectivity was also different than the bare and the Chitosan-Carbon nanotubes for each one of the molecules. While Chitosan-modified electrode didn't show any different selectivity to either dopamine or norepinephrine. So that's why we can use these multiple electrodes now to generate a set of cross-reactive electrochemical signatures in order to develop the chemometric model that will identify the fingerprint from the molecules. So we tested 100 different mixtures of different dopamine and norepinephrine in buffer. We divided them into training and testing sets, and we trained a chemometric model in an artificial neural network model to recognize the fingerprint of dopamine and norepinephrine in buffer in mixtures. And the results show that we get a very good prediction of both dopamine and norepinephrine, which had a percent correlation coefficient of 0.95, 0.94. So it gave us a good impression that we can use this as an approach to differentiate these overlapping signals. Now that was done in buffers, and the question is if we can do that, and we use this kind of system to measure directly in urine. So with that, we had a very enthusiastic graduate students that contributed, donated urine samples. We spacked them with dopamine and norepinephrine, did the same process now, trained a partially square regression model, and the prediction was very good when we were able to predict dopamine with a good percent correlation coefficient, 0.9. Norepinephrine was a bit worse, but still good, 0.79% correlation coefficient, and that was the first demonstration of the ability to predict both dopamine and norepinephrine directly in urine using this electronic tongue or bio-inspired approach. So with that, I would like to summarize and conclude. I hope I convinced you that it's important to rapidly analyze redox molecules or information, and it can be done with electrochemical sensors. I hope I showed you that convinced you that we can use modification coatings to directly measure specific redox analytes in biofluids such as the Clozapine detection in the blood. And I showed you a new type of approach using a type of a tongue approach to measure multiple redox molecules. This was done in norepinephrine and dopamine directly in urine. So I would like to thank my group, specifically to the people that work on this project were Rajendra Shukla, which is my PhD student, and my master students Alon Matsafi and Matan Arush. Also, I would like to thank to the funding agency for the Schizophrenia project, which is the Brain and Behavior Research Foundation, and for the urine approach, which is the Israeli Ministry of Science and Technology. Our collaborators, specifically for Professor Diana Kelly for the Schizophrenia from the Psychiatry Department and the University of Maryland, Baltimore. And if you're looking for more information, I'm glad to answer. And also, feel free to look at our website for any more information. And thank you all for listening.